Today we're going to talk about Section 3, Cell Boundaries. Our essential question is, what are the main functions of the cell membrane and the cell wall? Our objectives today are going to be to compare and contrast different modes of cellular transport. Please take notes during today's lesson and write down any questions you have while you're reading Section 3 of Chapter 7, and bring those questions with you to our live classes. Here's the vocabulary to be on the lookout in Chapter 7, Section 3. Feel free to pause this video at any time, jot down new terms, and I'll be defining each of these in today's lesson. A cell membrane is a thin, flexible boundary that surrounds a cell. Sometimes it's also called a plasma membrane. Keep in mind that all cells have a cell membrane. Some cells also have a cell wall, like plants, some protists, fungi, and bacteria, but not animal cells. In our previous lesson, we were making analogies for all the cell organelles and comparing a cell to a city. So if we continue to do that, if a cell is like a city, then the membrane is like the, and the wall is like the. So think about an analogy that would be good for the cell membrane and the cell wall during today's lesson. The functions of the cell membrane are to control what enters and what leaves the cell. It also provides protection and support. The structure of the cell membrane is a lipid bilayer, meaning it's a double layer of phospholipids. It's semi-permeable, which means it allows some materials to pass through and prevents other materials from passing through. Here's a nice picture of the cell membrane from your online textbook. Keep in mind here the cell membrane regulates what enters and leaves the cell. In addition to the phospholipid bilayer, we see proteins in purple and also some carbohydrates. Some of these molecules serve as markers, others are going to aid in cell transport, and we'll talk more about that today. Cell walls are found in fungi, plants, algae, and many prokaryotes, which are bacteria. They surround the cell membrane. Their function is to support and protect the cell, and they're usually made of some type of carbohydrate, such as cellulose. Here's some pictures of plant cells under microscopes. The cell wall provides a lot of structure, so plant cells under a microscope kind of tend to look like bricks of a building. Now we're going to start talking about how materials travel through the cell membrane. The cell membrane keeps a balance of dissolved molecules in liquid inside the cell and as well as outside the cell. Cells always want to maintain homeostasis, and they do that by having an equal concentration inside the cell as in their surroundings. And that concentration is a concentration of solutes. The concentration of solutes inside the cytoplasm is balanced with the concentration of solutes outside the cell. Cells will do just about anything to maintain this concentration. Concentration, again, is just the amount of solute. So for instance, the solute could be salt. The concentration could be measured in the mass of the solute or maybe the percentage of the solute. Diffusion occurs because particles tend to move from an area of high concentration to an area of low concentration. This doesn't require any energy, so we call this passive transport. Again, the cell's goal is to maintain equilibrium with its environment. Think of diffusion simply like if we put a lump of sugar in a beaker of water. As that sugar dissolves, the sugar molecules evenly distribute themselves in the beaker. This doesn't require any energy. They're just moving from an area of high concentration to areas of lower concentration. Osmosis is the diffusion of water across the semi-permeable membrane. Water will move across a membrane until equilibrium is reached. There's 3 ways we describe a cell or a solution surrounding the cell. Isotonic means that there's an equal amount of substances on both sides of the membrane, equal concentration of solutes. Hypertonic means there's more solute in one area than the other. And hypotonic means there is less solute. Let's look at some examples. In our first column, we see a cell in a hypotonic solution. In an effort to create equilibrium with its surrounding, the cell starts taking in water, trying to dilute itself. In the process, it could cause the cell to swell and possibly even burst. In the isotonic solution, the cell is already at equilibrium with its environment. Molecules are always moving, so water is still going to travel in and out of the cell, but at a constant rate, so we're not going to notice a difference in the cell size. In a hypertonic solution, the cell has been placed in a solution that has a very high solute concentration. In an effort to maintain equilibrium, the cell starts releasing water and shrivels up. Think very carefully about this and use this as a clue for this chapter's chapter mystery. Facilitated diffusion is basically diffusion with the help of a protein. Some molecules need help getting across the cell membrane, and proteins serve as channels for these molecules to do so. If we look here, we can see molecules that are too big to move through the phospholipid bilayer, and instead they're going through a protein channel. They're still moving from high to low, high concentration to low concentration, so this is not requiring energy of the cell. So we've just talked about 3 different types of passive transport. Take a look at these 3 pictures and try to match each picture with the type of transport we just learned. Pause this video if you need to review your notes. The boy up top on the slide is representing diffusion. He's moving from high to low. It doesn't require energy to do this. The picture on the left is representing osmosis. Here we have water moving from high to low. The water slide is representing the diffusion of water, which is osmosis. The picture on the right is representing facilitated diffusion. Think of a protein channel similar to a tube slide. Molecules are still moving from high to low, and this still isn't requiring energy. But the protein is helping out and serving as a channel for these molecules to travel. Next, let's talk about active transport. This is the process used to move materials across the cell membrane against the concentration gradient. So now we're moving from areas of low concentration to areas of high concentration. So think about those slides again. This would be climbing up the slide. Climbing up the slide is going to require energy, and it uses energy, the cell uses energy in the form of ATP, which is a high energy molecule. Proteins serve as protein pumps to pump molecules across the cell membrane. So here we have molecules being pumped from low concentration to high concentration, and energy would be required to do so. Here's a nice picture reviewing the differences between diffusion, facilitated diffusion, and active transport. Our top 2 are both passive because we're moving from high to low, and active transport is requiring energy because we're moving from low to high. There's a couple other types of active transport. These are most common in unicellular organisms as a mode of getting materials or food in and getting materials or waste out. Endocytosis is when a membrane changes shape to bring particles into the cell, and exocytosis, sounds like exit, is moving particles out of the cell. Sometimes we divide endocytosis into two different types, whether the cell is eating solids or if the cell is drinking liquids. So phagocytosis is when cells are eating solids, and pinocytosis is the cell drinking liquid. Here's a nice picture of an amoeba carrying out endocytosis. It changes its shape and surrounds that particle and literally engulfs that particle. All right, last review of today's lesson. Can you match the type of transport we discussed today with its definition? Pause this video if you need to. Again, active transport, movement of molecules from low to high, requires energy. Diffusion, movement of molecules from high to low. Facilitated diffusion, diffusion with the help of a protein. And osmosis, the diffusion of water. We learned a lot of new information in this section. Be sure to review Section 3 in your online textbook. Please bring any questions you have with you to our live classes, and I'll be available for help.